Yeah, so they asked me to chair the session, and most means I'll just be timing people, so we'll start with Josh first.
Thanks for the introduction. Thanks for the organizers for inviting me to come. I'm just going to briefly mention that some of the work I'm going to present was done by three very talented postdocs in my lab, Ali Mohabi, Arif Hamid, and Jeff Pettibone. And I'm also going to introduce or reintroduce the idea that there are different learning systems in the brain that operate using fundamentally different algorithms. I'm also going to totally ignore the hippocampus because no one cares about hippocampus. So the specific system that I'm going to talk about today is really basal ganglia centered and uses a reinforcement learning algorithm, and I'm going to talk about what that means. But I like the idea of introducing that there are different organization of circuits and microcircuit architecture that's specialized for extracting different features of your experience and processing them in different ways, specifically for reinforcement learning. And we've known, so one of the introductions to this field comes from maybe the 50s, and the discovery that there are certain places in the brain that you can wire up to a lever, and rats will rapidly learn to tickle their own brains in certain places and not others. And while there are many places in the brains that works, they're clustered around the mesolimbic dopamine system. And the key thing to remember about this situation is that rats will both learn to push a lever to tickle their own brains, and then they'll be highly motivated to go ahead and do it again and again and again, often at the exclusion of other activities. And I'm going to really go through the introductory slides fast because I'm sure most people, maybe not everyone, has heard about this projection, this famous projection from the midbrain, dopamine cell bodies sending their axons to various forebrain places, but especially the striatum. And famous work by Wolfram Schultz, which I promised Ila I would not spend more than a couple of minutes discussing, was really starting in the 80s and 90s, showed the following kind of phenomenon, that a dopamine cell, if you give an unexpected squirt of fruit juice into the monkey's mouth, will fire a burst of spikes. But as that fruit juice squirt becomes predicted, expected, the dopamine cell stops responding to it and instead responds to a cue that predicts that fruit juice. So even though the monkey still likes the juice and still avidly licks the juice, the dopamine cell doesn't fire here. So there, and it's also not just about making a prediction of future reward, it's about predictions in time. So this is tick marks of one dopamine cell firing, and if you delay the juice, you see a pause in firing, followed by a burst when the juice comes. If you bring the juice too early, you get the burst of firing early, and so forth. All right, so the current highly dominant paradigm in this field is that dopamine cells signal a reward prediction error. Talk about that. So if you're going to calculate a reward prediction error, you need a reward prediction. Right? And in turn, the point of a reward prediction error is to update your predictions. Right? It's a learning signal. It's there to do updating. And the way that this was conceived of by people, especially Schultz, was that there was this sort of globally broadcast signal from the midbrain to the forebrain that was really all one big signal, scalar signal. Right? And one reason why this has been such an influential and powerful paradigm, it explains all kinds of results at multiple levels of analysis. This is just one example from Kasai group looking at growth of individual spines on the dendrites of striatal neurons and showing that if you have this triple conjunction of glutamate uncaging onto that spine and postsynaptic depolarization and timed dopamine input happening together, then you can cause the spine to grow in real time. You can see it growing. All right? And the dopamine has to come in at just the right time to make this happen, within about a second or so. All right. So one of the reasons this has been such a successful marriage between neuroscience and computer science is that you can actually do useful stuff with reinforcement learning. And it's become extremely influential. A lot of the current developments in AI use reinforcement learning in one way or another. And some of the, especially in a computing institute, I'm not qualified to give a proper lecture on computing of reinforcement learning, but I'll just give some very basic ideas you need to know. So the idea of an agent that can be in one of a set of discrete different states, right? And each of these states has associated value, and a value is defined here as an expectation of future reward from that state, discounted in time, and I'll come back to that. Right, and if you transition between states to a state with unexpected value, then that jump in value is a reward prediction error, right? That sudden transition in values. So you're sitting around, and suddenly a cue comes on saying reward's coming up. Right, you jumped to a different state with a different value. That jump is the reward prediction error. Right, and then you use that reward prediction error to update the values of states or states and actions. Okay, And as I mentioned, this has become very powerful and useful and can do things like beat me at Go. And so one way of thinking about the brain, or at least this subsystem within the brain, that the cerebral cortex is there to represent states, like what state am I in, and to discriminate different states. And then the striatal and basal ganglia is here to paint those states with different values. Given I'm in this state, how much reward do I expect to get? And the point of the dopamine signal is to come in and modify the synapses here in order to modify and update the weights corresponding to values. All right?
Is there a better laser pointer?
You're going to ask me to do a two-handed bind mode. How can I use this one?
Oh, you're the better
Right.
showing.
Okay, now it's challenging, right? So there's a problem with this, which is this is a wonderful, absolutely wonderful framework, but it doesn't explain some of the core things we know to be true about dopamine, which is dopamine is not just there as a learning signal. Right? So there's lots of results that say dopamine also is motivating, energizing, invigorating. And it's really important to emphasize that everyone agrees that's true, but that is no part in this framework. Right? So something is missing. So what's this? We can talk about that. So one way of thinking about this question is you have an agent taking actions, interacting with the environment, and getting feedback about the environment. Right? So is dopamine fundamentally about this feedback, this learning, this updating role, or is it fundamentally about motivating, invigorating effects on online, ongoing performance? Right? Is that reasonably clear? And if it does both of those things, how does the system not get confused? So if you're a striatal neuron, say, listening to dopamine, we know dopamine can do lots of things to striatal neurons. It can affect synaptic plasticity, as I showed you, but also changes excitability of neurons, affects many ion channels. So how do you know whether to use dopamine as an invigorating, exciting signal, or as a synaptic plasticity signal, or what? Right? So the field has not really addressed this properly, but tends to do the following. It says, well, dopamine does both of these things, but on different time scales. Right? So fast, so-called phasic fluctuations in dopamine are supposed to be these reward prediction errors, whereas slower changes somehow do motivation. And no one who talks about tonic dopamine ever defines what timescale they're talking about. Right? They just say whatever timescale you're measuring with slow techniques like microdialysis. So a more specific set of ideas from Niv, Dor, and Diane is that the tonic dopamine level does not just motivation in general, but specifically signals the rate at which you're getting rewarded. This is a useful thing. If you're engaged in some activity and you're getting 3 rewards per minute, maybe that's worth continuing on more than some other activity that only gives you 2 rewards per minute. Right? So it's a useful decision variable deciding whether it's worthwhile to be engaged in some task or not. All right, so some of the data I'm going to present is trying to test some of these ideas and update them. And most stuff in my lab, not everything, looks like this. There's a rat in a box with some holes and some stuff in his head. And the task I'm going to talk about looks like this. A light comes on in one of those holes, and the rat has to stick his nose in it. And it takes him some time to go and stick his nose in the hole. That's lit up. I'm going to call that time the latency. All right? And then he waits there. And he's waiting for a variable period of time. And then he gets a go cue, which is just a white noise burst. And he pulls out, and he makes a choice. He can stick his nose in the left hole, immediately to the left or immediately to the right. And sometimes as he enters that side hole, he hears a click, which is the sound of a sugar pellet being delivered behind him. Right? And then he can go eat it if there's a sugar pellet. And it's a bandit task or a probabilistically rewarded task. And the probabilities for getting reward for choosing left or right change every now and then without telling the rat. He has to adapt his behavior accordingly. All right, so in this little sample of data here, a tall tick means he got reward, a short tick means he didn't. So he tried left, got a reward, tried left again, no reward, left again, no reward, let's try right, no reward, and so forth. And these are the nominal block probabilities here. So a full session of behavior might look something like this. These are the changing probabilities of reward, and I hope you can see with a little squinting that the rat is adjusting his choices between left and right to whichever option is better at a given time. But he's not just adjusting his left and right choices, he's also adjusting his motivation to play the game. So if we ignore whether it was left or right and just look at the density of rewards, sometimes he's getting a high density of rewards and sometimes a lower, sorry, I'm using the, should be there, sorry. Okay, now you can really see he's using a lower density of rewards and On the flipped scale here is a sort of latency. So when he's getting a high density of rewards, his latency to engage in the next trial is shorter. All right? This is one session of performance, easier to see this way.
Just a clarification
Yeah.
question. Is the reward, does the pellet remain in the bin until he harvests it? Or?
It does.
So then there is an advantage to going to the 10% bin, for example.
Oh, I'm sorry. So it's not the case that on each trial we give a reward whether he goes there or not, and then he can go harvest it. So he only gets delivery of the reward if he makes that choice.
Okay, so it's matching, not maximizing.
That's right.
Okay.
Take.
Yes. So yes, that would change the model, internal model the animal would use in interesting ways, and we actually are playing around with that. But this way it's very simple. So here's his adapting his choice. So as he enters a block of trials where left is much better, he switches his choices over to left and vice versa for right. And here's his vigor or latency data. So if he's got lots of rewards recently, 10 out of the last 10 were rewarded, he's really quick to initiate the next trial. And if he hasn't been rewarded any of the last 10, why should he bother? He's really slow to initiate the next trial.
Josh?
Yeah.
I'm confused by the ratios there. What's 90-50?
Sorry. Yeah, so 90-10 means that if he chooses left, there's a 90% chance he'll get a reward, and if he chooses right, there's only a 10% chance he'll get a reward.
What's 90-50?
90-50, yeah.
Oh, so it's 90% chance versus 50% chance.
Yeah.
Okay, so it doesn't add up to 100. Got it.
Yeah.
But following up on that, so 50-10 has a higher...
So if you compare 90-50 to 50-10, in both cases you're better off going left, right? But nonetheless, this one will have a higher absolute chance of getting reward, right? So this way it allows you to dissociate this process of making the better choice versus how valuable is that choice, right? So this kind of task has been used before to pull apart action values versus the choice itself. Okay, yeah, any more questions about the task? So, you know, I just showed you this thing saying that they're more invigorated or more motivated when they received lots of rewards recently, but how does it actually work? And you can look at the latency data and break it down a different way, which is to say that after the light comes on, what's the survivor curve or the hazard rate, survivor curve hazard rate, of initiating an approach behavior at each moment in time? And the way that reward history affects his decision making is it changes the instantaneous likelihood he'll decide to approach. All right, so higher reward history means a greater moment-by-moment chance. He'll say, okay, I'm going to make the decision to go for it. Does that make sense? So now we have this task where we have reward rates, history, and we have decisions to play the game, and we can see how does dopamine involved in all of this stuff. And we measure dopamine in too many different ways, 5, I think, 6. So I'm just going to start by talking about a couple of them. So we use microdialysis, which is historically a slow method, but has the advantage of it's much more specific to dopamine and to other things. And the fancy way we did it with Bob Kennedy was to measure lots and lots of things in the same 1-minute time resolution samples. Okay, so this is going to be our measure of tonic dopamine. And we also use fast-scan cyclic voltammetry, which is much faster, has 10 samples per second. It's focused on dopamine. It has its own idiosyncrasies. Like it usually doesn't work and stuff like that. We can talk about that. So, and this is just one example of one sample recorded from the ventral striatum here. And you can measure pretty much every major neurotransmitter at the same time within the striatum and all their metabolites. And you can then test various things. So does this concentration of each of these things in a minute-by-minute sample vary with things like the reward rate, right? So this is just a simple correlation between reward rate and dopamine. And you see, among all these things, only dopamine, and to a smaller extent the metabolite of dopamine, have this correlation with the sheer density of rewards that you're getting. So this is something that is indeed, seems to correlate with reward rate, and it's highly specific to dopamine. Right? And it's not about locomotor activity or explore versus exploit for those who are into these kinds of different ideas. If you just look at the total cumulative reward, which could correlate with time or satiety or any kind of things, lots of things care about that, but dopamine does not. Right? So an unpublished work, we have sort of extended this beyond the ventral striatum and looked at lots of different places in the striatum and medial frontal cortex. And what we find is that this correlation between reward rate and dopamine is seen specifically in the accumbens core here and in the ventral part of prelimbic cortex here. And if you go more dorsally or ventrally, in the frontal cortex you don't see it, and if you go more dorsally or ventrally in striatum you don't see it either. Right? So this is already a little inconsistent with the sort of idea of a broadcast dopamine signal. Something more specific is happening in specific subregions. All right? And this is sort of your map of hotspots of correlation between dopamine and reward rate. And one thing that I'm not quite sure how much weight to put on, but I quite like, is if you do like meta-analysis of human fMRI studies and say which parts of the human brain consistently correlate with value, subjective value, then you see two hotspots, which are in the ventral striatum and the ventral medial prefrontal cortex, which seems to correlate quite nicely to our rat hotspots. I think that's cute. So, so far this seems consistent with this idea that there's some tonic dopamine signal that can go up and down slowly and correlate with reward rate. But is it really what's going on? Is there really something slow going on that correlates with motivation and reward rate? And so we look at faster signals as well with voltammetry. And it looks like this is the rat performing the task. One of the holes is going to light up. He's going to stick his nose in it. There you go. And then he's going to make a left or right choice. He went left. All right. He heard a click. He can go eat a sugar pellet. And meanwhile, we're measuring sort of real-time patterns of dopamine release in the accumbens core here. Maybe watch another trial. So. Right. Okay, that time he didn't hear his click. Trying again. Okay. And then we can look at these fluctuations and align them on different events in the task, obviously. We can say, are there consistent fluctuations that correlate with light on or center in? And what you see is a fast ramping up of dopamine just as he completes his approach. And then if you break up the data by whether he got a reward or didn't, so at side in he either hears the click or he doesn't. If he does, dopamine goes up fast. If he doesn't, dopamine goes down fast. But the peak level of dopamine within a trial in this task is as he runs over to collect his reward, dopamine's getting up, 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 up. And then as he grabs it, dopamine goes down fast. And, obviously you can look at whether this pattern depends upon his reward history. And indeed, if he's gotten more rewards recently, then you see a smaller increase in dopamine. Sorry, wrong pointer. Right. And if he's gotten very few rewards, then the same click of sugar pellet delivery causes a bigger increase in dopamine. So this moment of time seems somewhat consistent with reward prediction errors, but other stuff happening in the trial, like as he approaches to begin the trial or as he goes to get his sugar pellet, don't seem consistent with this general idea of reward prediction error. Right? He's done this task tens of thousands of times. Why should going over to get his now totally predicted sugar pellet have any error associated with it?
Sorry, question.
Yeah.
So the undershoot that you see when he doesn't get a reward, that's essentially because it's been ramping just before?
Yes.
Or does it go below baseline levels?
Yeah. So when we first started trying to publish this data, we were very much thinking in terms of reward prediction errors. And one of the things that sort of bugged me about this is that it sort of seemed to go down and then stay down rather than bouncing back up nicely. And in fact, it goes down and stays down. And I cut out a few slides about that, but maybe I'll stick them in at the end. But basically, it doesn't really look like a reward prediction. All depends. Let me come back to that. If you're still interested in 10 slides, ask me. I'm trying to compress an hour-long talk into it. All right. So even though some aspects look like reward prediction errors, other aspects don't. Right? So, and this seems consistent with lots of other voltammetry studies. Right? So here's a fast ramp up in dopamine as a rat gets ready to push a lever for cocaine or for food and so forth. And here's another example from Gabriel and Paul Phillips showing a rat running down a maze arm to collect a fully predicted sugar pellet, and dopamine is ramping up as he runs down the maze arm. Right? So people have sort of contorted themselves trying to come up with computational counts in which this thing, which doesn't really look like a prediction error, really could be a prediction error. But I don't find any of those very convincing. Okay, so the story so far is we have these fast fluctuations in dopamine accompanying motivated approach behaviors that have some relationship to prediction error. And we have this microdallas thing about slowly measured dopamine correlating with reward rate. So are these all different things, or are they different aspects of the same thing? And obviously I wouldn't phrase it that way unless I thought they were all one thing.
Josh, can I just ask a question? So this is still in the context of the probabilistic task, right?
Yeah.
And so what you're saying is that, yeah, maybe this is a phase where there's been a lot of, sugar pellets at one of the ports, but nevertheless, there is a probabilistic element to it, right? So there's still some degree of surprise or, probabilistic.
In the task that we did. I mean, in some of the other tasks, like this one, there really wasn't any surprise associated.
And so then even so.
Blah, 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 blah. Okay, and so, in very brief fashion, I'm going to make the argument that this fast fluctuations in dopamine are not signaling reward prediction errors, they're signaling reward predictions in a form of a value function. And I think even David mentioned value functions yesterday. So what do I mean by that? This is probably obvious to some people, but imagine a sort of abstract situation where an agent has to pass through a sequence of states while remembering to use the right laser pointer. 1, 2, 3, 4, 5, 6. In order to get, and these are unrewarded states in order to get to a reward. Right? Well, it seems pretty obvious. It's better to be in state 8 than state 2 because state 8 is closer to reward. Right? So you can write the value of each state as a time-discounted expected future reward. Yes, good. All right, so the state value, the value being each state, is how much reward you expect to get, discounted by time. And in some ways, the point of a reinforcement learning algorithm is to use your ongoing experience to make estimates and adjust the estimates of how much reward you expect to get. Right? And this is useful because you have to make decisions. Is it worth engaging in some activity? Well, if you have a higher expectation of reward, obviously it's better to engage in it. So imagine a sequence where you're uncertain about whether you're going to get a reward. So this is your value function. You're on the 0.5 curve. And then you get a Q. And the Q says, aha, on this trial, you're definitely going to get a reward. Right? So your value of being in that state has just jumped up here. Right? And the amount of that jump is a reward prediction error. Right? So if you have a higher expectation of reward, and now you find out, yes, definitely going to get a reward, it's not very surprising. You jump up a little bit. You had a lower expectation. You get a bigger jump. Right? So these are so-called temporal difference reward prediction errors. They just are shifts in the value function. Right? So if you can keep track of value fast, you also get the reward prediction errors. And looks a bit like the dopamine data, but we could, the real task that we use is more complicated because there are variable delays and more events happening. So we made a model that evolves in real time, and we feed in the rat's actual data, behavior, into the model in order to generate real-time estimates of reward prediction errors and reward predictions. And it looks something like this. The particular set of states you use doesn't really matter. You just feed in the rat behavior, and you can do this. You can look at real-time dopamine fluctuations and say, do they look like the state values of the model that are evolving in real time, or do they look like the reward prediction errors? And the bottom line is, they look a lot more like the state values than they do like the reward prediction errors. Okay. So I just made the argument to you that if you keep track of value in real time, you can also use that to calculate reward prediction errors and use them for learning. But does the brain actually do that? All right? So one way we explore this is through optogenetics. So if dopamine is being used as a learning signal, then if you mess with dopamine fast, then what should happen is right now nothing. But next time you encounter the same situation, your behavior should be different. Right? That's what we mean by learning. Right? Whereas if dopamine is about motivation and online performance, then messing with dopamine should affect behavior right now. Right? And so we can look at that in the following way. So we can tickle dopamine cells and measure consequent dopamine release in the forebrain here, blah, blah. And we can give extra pulses of dopamine at different moments within the trial and see whether if we give dopamine at the same time as we turn on this light, is he faster to engage in an approach behavior, just as if we boosted his motivation, just as if we'd increased his expectation of reward, right? Or alternatively, if we give extra dopamine here, as he finds out whether he's going to get a reward, do we effectively boost a reward prediction error and reinforce his choice? So if he goes right, is he more likely to go right on the next trial? Okay. And in fact, both of these things happen. So if you look at the hazard rate of approach, so this is a latency and hazard rate. So giving a train of laser pulses immediately, within hundreds of milliseconds, affects his online motivated behavior. Makes him more likely to engage in an approach. Right? It's not a slow learning effect in any way. Conversely, we can reinforce his choices. So if we give a pulse of dopamine and reward feedback, then on the next trial, he's more likely to make the same choice again. This doesn't happen in littermate controls. We can make it go the other way with halo adoption and so forth. All right, so most of what I showed you so far is published. I showed you that reward, so that the dynamically changing dopamine signal seems to fluctuate to correspond to reward predictions much more than prediction errors. And that these dopamine signals used to make judgments, decisions about whether it's worthwhile working or not. That the fluctuations of this could be used as reward prediction errors. And we don't really believe in these different time scales of dopamine. We think there's just dopamine, which is fluctuating fast, and it can be used for both motivational and learning functions. Right. So, but we still have some problems. Right? We still want to account for the Schultz data, this kind of thing, saying that dopamine cells encode reward prediction errors, even though dopamine release seems to do reward predictions. So what's up with that? Right? So a couple of things we've been thinking about. One is that the dopamine cell recordings have almost entirely been performed in head-fixed animals performing Pavlovian tasks, where they really don't have anything to do except sit there and wait for rewards. So maybe the need to work is reflected in the dopamine encoding the value of work. Okay? And the other possibility is that dopamine cell firing and release are really different things, and both of these seem to be true. Okay. So one way we've been investigating this is by recording identified dopamine cells using optogenetics. And here's, just to sort of get consonance with the Schultz data, we also record the same cells in a more Pavlovian task. And if we give unpredicted food cues, these cells look like Schultz cells, more or less. They respond to unexpected rewards. Okay. And we can record the same cells in our bandit task and see what happens when you get reward cues or absence of cues. And the key things I want to point out are the following. So one is that these cells together do encode a positive reward prediction error, right? Not a negative reward prediction error for some reason, which is strange. We can talk about that. But the other thing is that the average firing rate or the tonic firing rate doesn't care about reward history at all. Right? So we have a situation where the tonic firing or the firing of dopamine cells has no relationship to reward history, even though dopamine release in the forebrain does. Right? So how do we score that? And one more example of the sort of dissociation between dopamine firing and dopamine release is seen here. So as I told you, the animals are sitting and waiting for the go cue, and they wait a variable amount of time. So as the animal waits, the dopamine cells start firing less and less and less and less. And this kind of thing has been seen before and interpreted as a negative reward prediction error. Right? So as you're waiting for the cue, your expectation that the cue is going to happen at the next moment is going up, 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 up. Right? Surely it's going to happen now, right? And so when it doesn't happen, you're more and more disappointed, right? It's more and more negative here. All right, just said that. So we can look at this either with recording individual cells or groups of cells. And in this fiber photometry experiment, we've been trying to simultaneously record dopamine cell bodies in the VTA and their terminals in the incumbents, and seeing if they're the same or different using GCAND. And using this method, we also see that the cell bodies are going down, 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 down during this delay period, possibly reflecting a negative prediction error. But the terminals of the same neurons are actually increasing their activity, more like a reward prediction. So we really think this kind of thing is showing us that control of dopamine terminals and release is largely independent of dopamine cell firing, and there's a whole field of biology that needs to be explored further here. All right, so just to wrap up, I just said this forebrain dopamine does not simply mirror midbrain dopamine cell firing. We think that local areas sculpt their own dopamine release, and we'd like to know how this happens. We'd like to know whether striatal neurons are switching modes, sometimes using dopamine as a learning signal and sometimes using it as a motivation signal. Are there many different functions of dopamine in online behavioral control, or is there one way that we can computationally bind these together? And the current working hypothesis we have is that dopamine is signaling how worthwhile it is to expend a limited internal resource, which could be energy or attention or time. So I'm going to stop there because I'm out of time.
A couple of minutes for questions.
It's a beautiful talk. I'm just worried, when I think of the basal ganglia, there's body configuration, there's a gin where the little mouse learns that 4 key presses is a macro. So I'm just wondering if that if the configuration of the body is a compound for some of your stuff. And also, this is a naive question, but I always thought there's lots of flavors of dopamine in there. And how do you know that you've got the right flavor?
Yeah, strawberry flavor. Maybe I'm not fully understanding what you mean by flavor. I mean, I understand that, like, you mean different.
D2, D3.
Sure.
Sorry.
So absolutely. So there are different receptors that listen to dopamine.
Yes.
Absolutely. And so I can certainly talk about how those receptors are listening to dopamine. There's been an idea that the sort of D2 receptors are listening to sort of steady levels of dopamine. There's some evidence that's true, some that it's not. But either way, whether you're a D1 receptor-bearing cell or D2, you still have this problem, right, which is what does dopamine mean to you right now? Okay? So you could be trying to separate out these by one set of cells cares about it as motivation, the other learning. But they both have to undergo synaptic plasticity. In both cases, they have the same fundamental synaptic dendritic architecture. They both have similar learning rules. So I think dopamine is modulating plasticity on both of those cell populations. So, yes.
How are you dissociating dopamine neuron firing and dopamine release?
How are we dissociating?
How do you imagine dissociating those two things?
You mean, how do I imagine that cells could... I mean, if you're a target cell at striatum, you have no idea what's happening with firing in the midbrain. You just know what the instantaneous concentration of dopamine is, right?
I thought when a cell fires an action potential, that action potential propagates along the
How old-fashioned of you.
Exactly. Could you bring me up to date?
Sure, sorry. So dopamine terminals have all kinds of receptors themselves, right? So for example, they have cholinergic receptors, nicotinic, and we know that even if you take a slice of striatum with no midbrain present, then if you flash acetylcholine optogenically, you get dopamine release, right? So there are lots of ways by which you can control dopamine release locally, and I should have been more specific about that, sorry. We also know, for example, that the basolateral amygdala input to ventral striatum can control dopamine release even if you inactivate VTA, right? So if you shut down VTA. So yes, there are mechanisms that have been known about for decades for local control of dopamine release. They've just never entered into the computational theories.
Does that mean then if you were to block local release, let's say by blocking, then you would still get the reward prediction aspect of this without the motivation aspect?
Yeah, well, we've been trying to find the right ways to get at that, right? So we've been trying, for example, to put carbon fibers for voltammetry on optic fibers for control of cholinergic cells and things like that. There's so much fast regulation in the system, that we don't even know the status of the cholinergic receptors, whether they're desensitized normally. So much is unknown about the status of these things in behaving animals that, and partly we're trying things and partly we're like, oh, it's too hard. Yeah.
I'm not quite clear. There's this dissociation between the release and the firing.
Yeah.
But there must also be some relationship there. So
Sure.
what does the firing contribute to the release?
Sure. So I actually think that the basic short story of a reward prediction signal is right? That you can use fast fluctuations as reward prediction error signals. But I think it's only half the story. So I think sometimes the striatum listens to this as a learning signal, and sometimes it uses a motivation signal. And I think, for example, the cholinergic interneurons are really important in switching modes, right? So, for example, the cholinergics usually fire steadily and then have brief pauses, a couple hundred milliseconds. Maybe Jun will talk about that, right? And then during those pauses, you unblock synaptic plasticity. Right? So there's evidence for switching into a learning mode very transiently and then switching back. And we think when you switch back, now you use it to gate excitability of the brain.
But then what does the firing contribute?
What is the firing of cells? Well, I think if you get a burst of firing of dopamine cells, you still usually get a pulse of dopamine release. Right? But it's a tricky deciphering job that has to be done at the level of the forebrain, and we only have the beginning of a sense of how that might work. Thank you.